This chapter starts our last major system of the semester, the nervous system. This particular chapter will deal primarily with neural tissue, but we will also do an overview of the nervous system and everything that we will be covering in these last several chapters. Neural tissue contains two types of cells. The neurons themselves, these are the cells that send and receive signals. They are considered to be the functional unit of the nervous system. There are also neuroglial cells called glial cells. These cells support and protect the neurons. They have a similar function as other types of connective tissue. The nervous system has two anatomical divisions, the central nervous system and the peripheral nervous system. Looking at the central nervous system briefly first, the central nervous system consists of the spinal cord and the brain. It also contains neural tissue, connective tissue, and blood vessels. The functions of the central nervous system include processing and coordinating. It helps to process and coordinate sensory data from both inside and outside of the body. It helps to process and coordinate motor commands. It controls activities of our peripheral organs, for example, our skeletal muscles. And it processes and coordinates higher functions of the brain, intelligence, memory, learning, and emotion. The other division is the peripheral nervous system. The peripheral nervous system includes all of the neural tissue outside of the central nervous system. Cranial nerves will carry signals to and from the brain, and spinal nerves carry signals to and from the spinal cord. The functions of the peripheral nervous system include the delivery of sensory information to the central nervous system and carrying motor commands to our tissues and systems. The peripheral nervous system is further broken down based on function. There are two functional divisions. The afferent division, afferent means sensory, information arriving, A for arriving. The afferent division carries sensory information from the peripheral nervous system sensory receptors into the central nervous system. So this is all information arriving at the central nervous system. This information is coming from interoceptors that monitor our internal environment, exteroceptors that monitor the external environment, and proprioceptors that monitor body position. The efferent division carries motor commands from the central nervous system to the peripheral nervous system. So this is information that is leaving or exiting the central nervous system. So E for exiting. So it carries this motor information from the central nervous system out to things such as our muscles and glands in the peripheral nervous system. There are two branches of the efferent, the somatic nervous system and the autonomic nervous system. The somatic nervous system controls voluntary and involuntary skeletal muscle contractions. The autonomic nervous system controls our subconscious actions, things that we don't have conscious control over, such as the contraction of our smooth muscles, our cardiac muscles, and our glandular secretions. The sympathetic division has a stimulating effect on the body, whereas the parasympathetic division has a relaxing effect. So the autonomic nervous system is further subdivided into the sympathetic and parasympathetic. Sympathetic revs you up, gets you going, and the parasympathetic calms you down. So this is a nice diagram that outlines the nervous system. So we said the two anatomical divisions, central nervous system, the brain and spinal cord, and the peripheral nervous system. The peripheral nervous system is broken down into the efferent and afferent. Notice the directions of the arrows. Efferent, exiting, motor commands, going out to the body. This includes the somatic nervous system that goes to our skeletal muscles, and the autonomic nervous system that includes the sympathetic and parasympathetic. The afferent is sensory, and this is information arriving into the nervous system from our various receptors. Now, most of this chapter is going to be dealing with nervous tissue. So we're going to start first by looking at the structure of a basic neuron. This is a classic neuron, the classic way we draw a neuron. The first structure are dendrites. These are these projections coming off. They're extensions of the cell body, which is this structure right here. Dendrites are designed to receive signals. This information comes in a one-way direction toward the cell body. So these pick up information and direct it toward the cell body. On the end of the dendrites, you may find spines. These help to increase the surface area available for picking up information. This is the cell body. It is also called the soma. This is the part of the neuron that looks like any other cell, classic cell in the body. This is where you find the nucleus and all of the other organelles. This long structure here is the axon. This is a long, slender process that sends out the signals. Again, it is a one-way flow from the soma out down this axon. The axon hillock, right here, is a thick section of the cell body that attaches to the initial segment of the axon. We can find collaterals. Collaterals are branches off of a single axon, and then they further branch into telodendria. These are extensions off of the distal end of that axon. And each of these telodendria end in a little knob known as the synaptic terminal. Now, neurons come in different shapes and different types. So we're going to look at the structural classification of the neurons. This one here is anaxonic. An means absence of an axon. So when you look at this, it appears that there is no axon, that it is all dendrites. These are very small neurons, and as I said, the dendrites and the axons all look the same. The majority of these will be dendrites, but one of them must be an axon. The way you tell is the direction of information. If the direction is coming toward the cell body, it is a dendrite. The one where the information is going away from the cell body will be the axon. These type of neurons are found in the brain and in our sense organs. Bipolar neurons are also relatively small. They will have one dendrite and one axon. They are found in our special sensory organs such as sight, smell, and hearing. So the axon is usually in the center with a dendrite on one side bringing information in and the axon on the other side of the cell body sending information out. Unipolar, they have very long axons. And the dendrites here will be on one end with a very long axon, and then the cell body usually is off to one side. These make up a good portion of the sensory neurons in our peripheral nervous system. And then the last is a multipolar neuron. This is the classic looking neuron. It has very long axon and multiple dendrites coming off of its cell body. So the dendrites, the cell body, and one long axon. These are very common in the central nervous system. They also make up the motor neurons that go out to our skeletal muscles. Neurons can also be classified based on function. We have sensory neurons. These are also called afferent neurons of the peripheral nervous system because they are taking signals into the central nervous system. Motor neurons are the efferent neurons of the peripheral nervous system. Efferent for exiting. They are taking signals out of the central nervous system to the effectors, such as our skeletal muscles. And then there are interneurons. Interneurons, they're also called association neurons. What they do is they connect one neuron to another, and they are only found in the central nervous system. The other type of cell we can find in nervous tissue are neuroglial cells. These are supporting cells of the nervous system, And there are many types in both the central nervous system and the peripheral nervous system. Those here in the yellow box, this represents the neuroglial cells of the peripheral nervous system. The ones in the purple box are found in the central nervous system. We're going to start with the cells of the central nervous system. The first are called ependymal cells. They form epithelium, also called ependymal, lining of the central canal of the spinal cord, as well as the ventricles of the brain. Their function is to secrete cerebral spinal fluid. They have either cilia or microvilli that help to circulate the cerebral spinal fluid, and they also help to monitor the composition of the cerebral spinal fluid. Next in the central nervous system are the astrocytes. They have projections coming off of them that wrap around capillaries to form the blood-brain barrier. They help to isolate the central nervous system and control the interstitial environment. The way they're arranged, they will create this three-dimensional framework for the central nervous system. They are also involved in repair and can help repair damaged neural tissue. And they also help to guide neuron development during embryonic development. Next in the central nervous system are the oligodendrocytes. They have projections that they send off to wrap around sections of axons to form a myelin sheath. One oligodendrocyte can wrap itself around many axons and make this myelin for many axons all at the same time. This myelin sheath helps to insulate the axons and prevent erroneous signals. Myelin also helps to speed up the rate of propagation of a signal. And the presence of myelin on an axon will give that nerve a white color. The last one in the central nervous system are the microglial cells. They are capable of migrating within neural tissue. They are the cleanup cells and the cells for protection. They can engulf waste, debris, and pathogens that might be present, and they do this via phagocytosis. So here's a summary picture. See how well you can identify the different neuroglial cells. Here's A. It's coming out, and notice it's wrapping its projections around the axon of a neuron. That is an oligodendrocyte. B, these are the last little ones we talked about, the microglial cells. C, it's wrapping around a blood vessel. This is an astrocyte. And the last one here is forming the lining of that canal or that ventricle. Those are the ependymal cells. Switching to the peripheral nervous system, we can find satellite cells. They help to regulate the environment around the neurons. They have a similar function to the astrocytes in the central nervous system, and they surround ganglia. Then the other one we have are Schwann cells. They are responsible for forming the myelin sheath around our peripheral axons, very similar in function to the oligodendrocytes. However, one Schwann cell can only produce myelin for one segment of one axon. It takes many Schwann cells to create the myelin sheath for an entire axon. Also, Schwann cells do not always result in myelination. When we describe our peripheral nerves, we describe them as being either myelinated or unmyelinated. What does this mean? This picture here illustrates a myelinated axon and how it forms. When an axon is myelinated, that Schwann cell will wrap itself, its membrane, around and around and around that axon many, many times. This produces a thick layer around that axon. And also notice in this picture, I said it takes many Schwann cells to produce the myelin for a single axon. On this side, we see an unmyelinated axon. One way to picture this, think of an axon like a bare wire. You know, in a cord, if the plastic insulation on the cord becomes frayed and those bare wires are exposed, that can be a problem. It can short circuit out. It can create a spark. The wires need to be insulated. Our axons and our neurons are no different. So in an unmyelinated axon, what it's doing, one Schwann cell can grab a hold and separate several axons. It is not wrapping itself around that axon. It is just separating them and insulating them and preventing bare axon from touching bare axon. So we haven't produced, you can see it here, those are the axons. There is not a thick coat around it. This is the nucleus of the Schwann cell. Over here, here's the axon, and look at that thick coat around that axon. Neurons or nervous tissue will respond to injury, but it is different whether we are in the peripheral nervous system or the central nervous system. In the peripheral nervous system, the process is called Wallerian degeneration. What happens? Here, there's been damage to the neuron. The axon has been severed. The axon distal to the site of injury will disintegrate and degenerate. Macrophages will come in and start to clean up the debris. Schwann cells, however, remain, and they will form a new tube for growth. The damaged end of the axon also, notice, it begins to sprout these little buds. And these buds will begin to grow into this tube that was created by the Schwann cells. As that bud is growing, it will be wrapped in myelin as it progresses down the tube. This is an extremely slow process, taking the axon grows less than a millimeter every couple of days. In the central nervous system, there is a much more limited ability to repair. The astrocytes release chemicals that will inhibit the growth of the damaged neuron. And astrocytes also form scar tissue that act as a physical barrier. So even if the neuron did sprout a bud, it would be very difficult for it to grow. Plus, the oligodendrocytes do not form a tube. So there's no path for that growing neuron to follow. Now we're going to get into how neurons function. Neuron carries information, and all of this information travels as an electrical impulse. This electrical impulse is known as an action potential, and it moves down that axon. As an action potential moves, the intensity of that impulse does not diminish. It stays at a constant level all the way down that axon. The impulse, this action potential, is based on movements of ions across the cell membrane. And this movement is set up by the sodium-potassium exchange pump. Now let's review this pump. We talked about this back in Chapter 3. Remember, this is an active transport pump, which means it requires ATP in order to function. And what it does, for every 1 molecule of ATP, it will pump 3 sodium ions out of the cell and at the same time bring 2 potassium ions in. Now notice, both sodium and potassium carry a positive charge. So what's happening, we are pushing 3 positive charges out of the cell, but only bringing 2 positive charges back in. This is going to create what is called a transmembrane potential. The inside of the cell is more negative compared to the outside because more positive charges are pumped out compared to those brought in. This creates a charge, and this charge is a minus 70 millivolts in most cells of our body. This is an important number to remember. As a result, we create this electrochemical gradient. Let's describe what's happening. The top picture looks at sodium. Now remember, where is there more sodium, inside or outside of the cell? The bulk of the sodium is outside of the cell, and there's very little inside. So there is a chemical gradient, okay, based on concentration gradients that sodium wants to come in. to achieve equilibrium. But remember, we can't have that. The inside of the cell has a negative charge. The outside is more positive. Sodium has a positive charge. That creates an electrical gradient. You put those two together and you create this very strong electrochemical gradient that is trying to pull sodium in. So sodium wants to enter the cell. Looking at potassium. Most of the potassium is inside of the cell, very little outside. So the chemical gradient is going out to try and achieve equilibrium. The inside of the cell is negative. Potassium has a positive charge, so the electrical gradient is drawing potassium in. Put these two together, it creates less of a push for potassium out of our cell. Potassium wants to exit the cell. But again, remember that we don't want this to happen. We want to maintain this difference on either side of the membrane. So when these ions move across the membrane, it will change that transmembrane potential. So if positive ions enter the cell will become less negative and move closer to zero. If a positive ion leaves the cell will become more negative and move even further away from that -70. So let's look at some important numbers relating to this transmembrane potential. The resting potential. This is the transmembrane potential of a resting cell. As we said before, this number is -70. Our cells, neurons, are always being bombarded with different signals, and they will create a graded or a local potential. So a stimulus can produce a temporary change in the resting potential. These stimuli can cause small influxes of sodium, not enough to create an action potential. A graded potential is not an action potential. A graded potential is just a slight change in that transmembrane potential. Maybe we go from -70 to a -65, okay? Slight changes. An action potential occurs when the electrical impulse moves across that cell membrane. It is caused by a graded potential that reaches another magic number that we call threshold, and that is -50 millivolts. We have to have a strong enough signal to allow enough positive charges to come into the cell to raise the charge to a -50. Then we will get an action potential. Now these two numbers are important, -70 and -50. Unfortunately, your book changes these numbers from picture to picture. So for our purposes, the transmembrane potential will always be -70. Threshold, the number we need to get to generate an action potential, will be -50, irregardless of what the picture shows from your textbook. Now, in our cell membrane, there are various channels, and the transmembrane potential will rise or fall in response to temporary changes in the permeability of the membrane. This change in permeability results from either the opening or closing of specific channels for either sodium or potassium. Now, these channels in the membrane are either passive or active. Passive channels are called leak channels. They're always open, and this is how sodium and potassium can kind of sneak across the membrane. It makes the membrane leaky to both of these. And also their permeability and how leaky they are can change under different conditions. The other channels are active channels. They are also called gated channels because they have a gate that will keep them closed. At the resting potential of -70, most gated channels are closed. Now, different conditions can happen to change these channels. So at any given moment, a gated channel may be closed and not capable of opening. It is inactivated. Think of it as the gate is locked and nobody has the key. So you're not getting in, you're not getting through. Closed, inactivated. Or it could be closed but capable of opening. Here, yes, it's closed, the gate is closed, but it's not locked. So it's easy to open this gate. And lastly, open or activated. And now that gate is wide open and ions can pass through. All right, we need a stimulation to generate an action potential. What a stimulation does is it will open some of the gated sodium potentials, depending on how strong that stimulus is. Here, we're looking at just a graded potential. We had a little small stimulus that opened a few of those sodium gated channels, and sodium came in. And the transmembrane potential rises. But notice it only went up to -65. This is not going to travel down that axon. It's just a change in that potential at this site where the stimulus occurred. Because the transmembrane potential moved closer to zero, we say the neuron has been depolarized. Depolarization is a shift in the transmembrane potential closer to zero. Now, in order to get spread of the impulse down the axon, we need a stronger current. We need enough sodium ions to come in, and here's where change this number to -50. We need enough sodium to come in to change that potential to threshold, the magic number. And what that will do is open up the next series of gates in line. And the sodium starts to move down this axon, opening gates along the way. And this is how an action potential is going to move. Now, what happens after a graded potential? This is not an action potential. This is just these slight changes in the membrane. We have to repolarize. So what happened? We have a stimulus was applied. We depolarized. We moved closer to zero. When that stimulus is removed, that transmembrane potential needs to go back to normal, back to resting. Sometimes, though, a stimulus will take that transmembrane potential in the opposite direction, further away from zero. This is known as hyperpolarization. We are going to increase the negativity of the resting potential. This happens when potassium channels are opened and potassium starts to leave the cell and the cell loses more positive charges. It has the opposite effect of opening a sodium channel. All right, now let's focus on action potential. An action potential is a change in that transmembrane potential that now can spread along that membrane. An action potential needs a graded potential large enough to reach threshold. This is this should be somewhere around, again, it should be -50, not -55. An action potential is an all or none principle. An action potential will either be triggered or it won't. As long as that stimulus is strong enough to reach threshold, an action potential will be generated. The action potential will be the same in that neuron, no matter how big the stimulus is. So, for example, if you stimulate the axon with 10 volts, you get an action potential. But if you stimulate it with 50 volts, the action potential is going to look exactly the same. As long as you reach threshold, you will get an action potential. Now let's look at the steps involved in an action potential. Here's our resting state. We have to depolarize that membrane to threshold. And again, change this number to 50. So we have a graded potential that is strong enough to open up enough gates to raise that transmembrane potential to threshold of -50. This will then begin to activate sodium channels. The sodium voltage gated channels will open up and sodium will begin to rush into the cell. This is going to result in a very rapid depolarization. Look what happened to our charge. We're up to a plus 10 millivolts. Sodium has rushed into the cytoplasm, and that transmembrane potential changes from a negative to a positive. Next, we are going to inactivate those sodium channels and activate the potassium channels. Potassium is also a volta-gated channel. It opens at a plus 30 millivolts. So the inactivation gate closes so that sodium channels are inactivated. It is impossible now for any more sodium to enter the cell. These potassium channels, once they open, they will allow repolarization. Okay? Potassium is going to start to move, and our charge is going to start to drop again. We have to come back to our resting potential. The problem is, potassium channels are very slow to close. When the membrane potential reaches the resting potential of minus 70, those potassium channels will start to close. But it takes them a while, and while they are closing, even more potassium can leave the cell. This results in the membrane being hyperpolarized down to a minus 90 millivolts. That's okay. Our sodium-potassium exchange pump is there, and it will return this membrane back to its original minus 70. At this point now, the action potential is over. This is another way of looking at the different steps. So again, at rest, okay, we are at minus 70 millivolts, the transmembrane potential, and both gates are closed. They're not locked, they're just closed. When we hit threshold, which should be minus 50, okay, sodium channels are still closed, and so are the potassium. It takes a little bit more to push those gates wide open, and then sodium starts to rush in, and look what happens to our charge. Once that charge reaches a plus 30, those sodium gates will be locked now, and the potassium gates open. And we come back toward our minus 70. The potassium gates do not close until we hit minus 70. And like I said, it takes a while for them to close, so we tend to hyperpolarize. We go to a minus 90. This will then be fixed back to the minus 70 with our sodium-potassium pump. Now, the cell needs and takes some time to reset before a second action potential can happen. Remember, action potentials start by opening sodium channels. So if the sodium channels are already open, or if those sodium channels are locked, there is no way to generate another action potential. These are known as refractory periods. There is an absolute refractory period here where no action potential is possible. Absolutely not. Why? Because the sodium channels are either already open or they are inactivated and locked. Out here, this is known as a relative refractory period. We could get an action potential, but we would need a very strong stimulus in order to do it. The reason is we are further away from threshold, and we have to work against the flow of potassium during this period. Now, let's look at a couple of things related to this. Local anesthetics, for example, drugs like Novocaine. How do they work to block pain? What they do is they lock the sodium channels in an inactivated state. So by doing this, you prevent any action potentials in that nerve cell. If an action potential cannot travel down that neuron, you will not feel pain. But you need to be careful. Local anesthetics must never be given systemically because they are very indiscriminate. They will block every sodium channel they come in contact with. So they must never enter the brain or the heart, or with the heart, it could stop the heart from beating. Let's look at how an action potential is spread, or the propagation. The speed of an action potential along a membrane. This is propagation. Remember that the axon, or the action potential, starts at the axon hillock, and then it will move down the entire length of an axon. This represents a series of repeated actions, not passive diffusion. There are two methods for propagating an action potential. Continuous propagation, which happens on unmyelinated axons, and saltatory propagation, which occurs on myelinated axons. Let's look at continuous propagation first. Again, this is what happens in unmyelinated axons. Here, this will affect one segment of the cell membrane at a time, all the way down that axon. So what happens? You will get an action potential in that very first segment, depolarizing the membrane to a +30, and this will generate a current that depolarizes the second segment to threshold, developing an action potential. When the second segment is generating an action potential, that first segment will be in a refractory period and cannot be re-stimulated again. This creates a current that will depolarize, keep depolarizing the next segment in line. Now, this is supposed to be a little video to show this, but it doesn't work, or it's not supported by newer systems of PowerPoint. But you can see you have to change this potential all the way down that membrane. This is a very slow process compared to our second type of propagation, saltatory. Saltatory occurs in myelinated axon. It is much faster and uses less energy than the continuous form. Myelin, remember, insulates the axons and prevents continuous propagation. When the Schwann cells myelinate the axon, they do not cover the entire axon. They leave these little gaps in between the Schwann cells. These are called nodes of Ranvier. So what happens in saltatory conduction or propagation, you will generate an action potential at the node. And this will open channels. Sodium will move down through the cytoplasm, through that insulated area to the next node. Open sodium channels here. They rush in, move down, and open the next. So it creates this current that jumps from node to node to node. So depolarization only occurs at the node, and as a result, it can move much faster. Information, remember, travels as action potentials, and all action potentials look the same. They're electrical signals. The most important information, such as vision, balance, etc., must be carried quickly. How is this done? The larger the axon diameter, the faster the speed of propagation. And when an axon is myelinated, this will create faster propagation. Let's revisit the synapse. We looked at this at the neuromuscular junction earlier, but a synapse isn't just between a neuron and a muscle cell. It is between a neuron and any other cell. So yes, there is the neuromuscular junction, but there are also neuroglandular junctions. The terminology that used, we have a presynaptic cell. This is the neuron that sends the message. Presynaptic, before the synapse. We have the postsynaptic cell. This is the cell that receives the message after the synapse. And then we have the synaptic cleft. This is the gap that separates the two membranes. Now, we've said before that a synapse is an almost connection. In this example here, the two cells do not touch. But that is not the case for all synapses. Another type of synapse is an electrical synapse. Here you will have direct physical contact between the cells. The cells communicate through gap junctions. These gap junctions will allow ions to pass between the cells. This will produce a continuous local current and action potential propagation. Electrical synapses are seen in areas of the brain, the eye, and other various ganglia. The majority of the synapses, though, are chemical synapses, where the cells are not in direct contact. Here, neurotransmitters will carry the signal across that synaptic cleft. Now, it's not guaranteed, just because a neurotransmitter is released, it is not guaranteed that an action potential will happen in that postsynaptic cell. An action potential may or may not occur in that postsynaptic cell. It depends on how much neurotransmitter is released. Remember, we have to have enough neurotransmitter to reach that threshold of the postsynaptic cell. It also depends on the sensitivity of the postsynaptic cell. Now, neurotransmitters, they are released by the presynaptic neuron. They are chemical messengers. And what they do, they're released by the presynaptic membrane, but affect and bind to receptors on the postsynaptic membrane. And remember, they are very quickly broken down by enzymes. We talked previously about acetylcholine. This is the neurotransmitter at the neuromuscular junction. And remember, it is quickly broken down by the enzyme acetylcholine transferase. Let's look at the process of a synapse. Okay. This is going to be, I forgot. There are two types or two classes of neurotransmitters: excitatory neurotransmitters and inhibitory neurotransmitters. An excitatory neurotransmitter will cause depolarization of the postsynaptic membrane, and they will cause an action potential. An inhibitory neurotransmitter will cause hyperpolarization of the postsynaptic membrane, and this will suppress an action potential. The effect can also be varied by the type of receptor that receives the neurotransmitter. Acetylcholine, for example, results in stimulating muscles, so it is excitatory for our muscles, but it is inhibitory if it binds to receptors in the heart. All right. Sorry for the backtrack. Let's look at the steps in a synapse. Step 1 is the action potential arriving at the synaptic knob. And remember, the neurotransmitter is stored in vesicles in this knob. This action potential, as it comes down, will result in depolarizing this synaptic knob. As a result of being depolarized, calcium enters into from the synaptic cleft, will enter in, triggering the release of acetylcholine. This depolarization of the synaptic knob opens up calcium-gated channels, allowing this calcium to come in, and this is the signal for the release of the acetylcholine. The release of acetylcholine, though, will stop very quickly because calcium will be rapidly removed from the cytoplasm of the synaptic knob. It doesn't stick around very long. Acetylcholine will now cross the synaptic cleft and bind to receptors on the postsynaptic membrane. This will cause that postsynaptic membrane to become depolarized. These are sodium-gated channels, and therefore it increases the membrane's permeability to sodium. This is a graded potential, which means the more acetylcholine that is released, the greater the number of channels that will be opened, and the more the sodium rushes in, the more that membrane will be depolarized. If the depolarization brings that membrane to threshold, an action potential will occur. And you can see here it was, and that action potential is moving down the membrane. The last step, acetylcholine will be removed by acetylcholine esterase. The effects on the membrane is only temporary due to this enzyme. Most of the acetylcholine is broken down actually before it can even reach the postsynaptic membrane. Now, every time you have a synapse, there is a delay in the transmission of that information, anywhere from 0.2 to 0.5 milliseconds. So this delay is between the arrival of the action potential at the synaptic knob and the effect that it will have on the postsynaptic membrane. This reflects the time for the calcium influx and the acetylcholine to be released. So the fewer the synapse that information has to cross, the faster your response is going to be. We are going to look at reflexes in an upcoming chapter, and we will see the effect of multiple synapses. Neurons can fatigue. Neurotransmitters sometimes cannot recycle fast enough to meet the demands of a very intense stimulus. The synapse will be inactive until the neurotransmitter is replenished, and that nerve will be fatigued. Now let's look at some other important neurotransmitters. Yes, we've looked at acetylcholine, and we know it is excitatory when it binds to receptors on our skeletal muscles, but it is inhibitory when it binds to the cardiac muscle. Norepinephrine, this is excitatory in both the brain and the autonomic nervous system. Dopamine can be either excitatory or inhibitory. For example, it has inhibitory effects that play a role in the control of our movements. Parkinson's disease has been linked to a decrease in dopamine levels. What's one of the classic symptoms of Parkinson's? It is the shaking. That shows the effect of dopamine. It helps to control the movements. Serotonin is excitatory in the central nervous system. It has an effect on emotion and attention. And also in the GI tract, it is excitatory. Decreased amounts of serotonin have shown to result in depression. And gamma-aminobutyric acid. This has an inhibitory effect in the central nervous system. It helps to reduce anxiety. Anti-anxiety drugs will cause an increased production of GABA. Now, along with neurotransmitters, there are also neuromodulators. They too can be released from the synaptic knob. They are similar in function to neurotransmitters. However, their effects tend to be longer and they are slower to appear. The response to a neuromodulator involves multiple steps, and they can affect the presynaptic, the postsynaptic membrane, or both. So neuropeptides tend to bind to receptors to activate enzymes. An example of a neuropeptide are opioids. These are found in the central nervous system. These are naturally occurring pain relievers, such as these are endorphins and enkephalins. Opium is named after this, opium and morphine. These are drugs that work very similar to these natural opioids. Now we're going to look at how neurotransmitters and modulators can work at our cells. There are two different effects they could have. One is a direct effect, and this is how the neurotransmitter acetylcholine works. Acetylcholine exerts its effect by directly opening or closing a gated channel. So yes, acetylcholine works this way, and then glutamate and aspartate, which are other neurotransmitters, work exactly the same way. Others work through an indirect effect. So they work through intermediates called second messengers. So the neurotransmitter, it will be our first messenger. Okay? It's going to deliver the message to receptors on the membrane. And then the second messenger will be either ions or molecules that are either produced or released inside the cell as a result of the first messenger. It is the second messenger that ends up opening the ion channels. So this is an indirect. The neurotransmitter indirectly opens those channels through a second messenger. The most common second messenger is cyclic AMP. This is how epinephrine, norepinephrine, dopamine, many of these neurotransmitters work. This takes a little bit longer because we have to rely on the second messenger to open that channel. Now, the actions of these neuromodulators and neurotransmitters and hormones, they can change the membrane sensitivity to neurotransmitters. They can result in shifting the balance between excitatory stimuli and inhibitory stimuli. For example, morphine reduces the effect of an excitatory stimuli on the pain processing centers in the brain. How do our neurons process all this information? Many dendrites are receiving neurotransmitter messages simultaneously, all at the same time. Some of this information is excitatory and some is inhibitory. So how does it know whether or not it's supposed to send that information on? The net effect at the axon hillock will determine if an action potential will be generated. A neuron is said to be inhibited if the transmembrane potential is lowered away from threshold. In other words, becomes more negative. So when that neuron adds up all that information, it makes it even more negative. That neuron is inhibited and no action potential will be set. Facilitation means the transmembrane potential has been raised closer to threshold. So in other words, when it adds up all that information, we've raised it to a -60. Oh, we're close. We need to be at a -50. So that neuron is facilitated. It will not send an action potential, but it's closer to being able to do so. One way we can reach, our neurons reach an action potential is through a process called summation. Here, many of these sub-threshold excitatory stimuli can be summed up or added together to push it over and push it up to that -50. There are two ways for summation. Temporal summation. This is when a neuron is bombarded multiple times in a row. Rapid, repeated stimuli at a synapse that eventually opens enough channels to bring that cell to threshold. Spatial summation. Here we have two different signals coming in, multiple locations that are sending information. So many stimuli arrive at multiple synapses, and when you add up the effect, it is enough to reach threshold and send an action potential.